Hi, in this video, we're going to learn about DNA analysis, which is a part of week 9 lab. So let's understand about the outline of this presentation. First, I'll introduce you to DNA analysis by talking about the definition and why do we do it. Next, I will talk about the structure and function of DNA. Then we will refresh our memory from our previous lab on genetics, just to see how DNA analysis is linked with the genetic variations and other inheritance patterns. Then we'll understand the key methods or the main methods of DNA analysis. And lastly, the applications. So let's start with the introduction. DNA is also known as deoxyribonucleic acid. It's a long name, but it's pretty easy, deoxyribonucleic acid. It carries the genetic instructions for growth, development, and functioning of all living organisms. By analyzing the DNA sequences, scientists can identify individuals by forensic analysis or paternity tests. They can diagnose genetic disorders or inherited diseases. They can study ancestry and evolution through, by tracing lineages. So the definition of DNA analysis is that it is a process of examining and identifying the DNA sequences to understand genetic information. Moving on to the DNA structure. It consists of 3 basic components. First is the phosphate group, which is this group, the phosphate group. It provides negative charge because of the negative O over here, oxygen, negative charge to the entire DNA. It forms the backbone by linking to the sugar molecule of another nucleotide, and also to its own nucleotide. So over here, it will be linked to some other nucleotide, sugar molecule, and this is its own DNA molecule. So there'll be a series of sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way in the DNA structure. Next is the sugar, which is deoxyribose. So deoxy means it has 1 less oxygen in it. And it is a 5-carbon ring. So you can see here, it is 1, 2, 3, 4, and 5. Opposite, 1, 2, 3, 4, 5. Yeah. So sugars are 5-carbon rings, and it connects phosphate group and the base. So this is the nitrogen base, and this is the phosphate group. It connects between the two. And the third one is nitrogenous base. Any one out of these 4 bases will be present in one particular nucleotide. So these 3 components make up one nucleotide. A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. Now, DNA is a double helix structure, and this is a base pairing rule which is going to be consistent in all types of organisms. A will always bind with T, and G will always bind with C. So over here, you can see that A and T share 2 hydrogen bonds, and G and C shares 3 hydrogen bonds. So this base pairing rule is consistent throughout. And the next is that they have anti-parallel orientation. So 5' to 3' and 3' to 5'. If you see here, the dash over here or the apostrophe is known as prime. You call it 3' and 5'. It's going to be consistent. So one strand will be 5' to 3', and next strand would be opposite, 3' to 5'. And that's also consistent throughout. This is just an overall diagram of how the different A and T, G and C molecules are connected to each other. So if you see thymine and adenine, they share 2 hydrogen bonds, and guanine and cytosine share 3 hydrogen bonds. Genetics refresher. So genetics is the study of how traits are inherited through genes which are made of DNA. Genes are the segments of DNA that contain instructions for building proteins and other building blocks of life. Chromosomes are long DNA strands packaged tightly with the nucleus of the cell. Humans have 23 pairs of chromosomes. Next is traits. They are passed on from parents to the offsprings through genes, and each parent contributes to 1 copy of each gene in the child. Alleles are different versions of a gene which determines the variations in traits. For example, eye color has a separate set of alleles. Similarly, your height has a different set of alleles responsible. Moving on to dominant alleles and recessive alleles or traits. Dominant alleles mask the effects of recessive alleles. So if you have 2 dominant alleles, it will be expressed. If you have 1 dominant and 1 recessive, the dominant trait will be expressed, and the recessive alleles will be expressed only if you have homozygous recessive. So if both the alleles are recessive, only then the recessive alleles will be expressed. Genotype versus phenotype. Genotype is the genetic makeup of an organism, which is the letters that we use, capital A, heterozygous AA, or homozygous lowercase a. And phenotype is the physical expression of a genotype. So what you see morphologically is going to be the phenotype, blue eyes, or like the blood type, or the height, etc. So this is the overview of the entire process of DNA analysis from starting to end. How you collect your samples, it could be your blood sample, saliva, semen, touch, like a fingerprint, cigarette butts, hair, nails, tissues. And how you take these samples is by a swab, like what we did for COVID testing, make a cut, maybe a punch, or just tape it down with like a fingerprint for fingerprints. Then you perform a direct PCR. There are multiple types of PCR, which you don't need to get into right now, but just understand the overall process of what a PCR is. We'll talk about it later. So PCR is polymerase chain reaction, and it's basically amplifying or multiplying the small segment to make either a lot. So it's basically a small DNA fragment multiplied by 30 or maybe 40, depending upon what you choose. So you'll have that many copies of that small strand of DNA. So you're amplifying that small strand so you can visualize it further. Next step is gel electrophoresis. So after amplifying, you want to check what is the connection between the different DNA samples. What is the difference in sizes or presence of certain genes, etc. That is DNA analysis, which means which is what scientists use to maybe solve a criminal case. The first part, which is direct PCR or just PCR in general. PCR, as I mentioned, it is polymerase chain reaction. If you see here, okay, first the definition. It is a technique used to amplify or magnify the specific segments of DNA. It allows scientists to produce millions of copies of DNA sequence from a small initial sample. The components of a PCR mix or a PCR reaction are first a template DNA, which means which is the initial sample that you're magnifying. Primers are the small starting material that you add to the mix so that the template will, this is the template, this is the primer, it will attach to the template, the starting point of template, and it will add more nucleotides to it. So DNA polymerase is the enzyme which will add more nucleotides to the same DNA strand. And from where will they get the nucleotides? So you add the nucleotides individually in the form of DNTPs, that's deoxynucleotide triphosphates. Yeah. And buffer solution to maintain the correct temperature conditions or pH conditions in the entire PCR mix. So again, very briefly, components are template DNA, which is the DNA strand or DNA portion that you want to amplify or magnify. Second is primers, which is the small starting material of the same DNA, which will be complementary. So if the initial strands or initial nucleotides are ATGC, then the primers, like the short primer that we attach to this, which will be complementary to the initial strand, will be, so ATGC, that will be TACG. So the primer will be TACG and the DNA strand will be ATGC, so that it can bind or connect with the hydrogen bonds together. And then DNA polymerase is the enzyme that will add the nucleotides present in the mix, which is the DNTPs. And buffer is to ensure that the conditions are well maintained. The process, if you look at the process, so this is the template DNA strand, 3' to 5' and 5' to 3'. Original DNA to be replicated is the template DNA. This is the template. The red portion over here is the primer or the starting material. The very first step is to denature the DNA strand. So DNA is going to be double helix structure. You need to separate it first by heating it. So heating will separate the two DNA strands into individual strands, like single-stranded DNA molecules or DNA strands. Now, once they have been separated, the primers will act on them. And primers will attach to the DNA strand individually, like both of them. And that step is known as annealing. It happens at a specific temperature, and it's consistent between all the reactions, or not. Yeah, it is consistent between all the reactions. So denaturation is separation of the initial DNA strand into two. Annealing is the step in which the primers bind to the individual separated DNA strands. And third step is the elongation, where DNA polymerase adds the DNTPs individually to the DNA template in the same direction as they were generated. So this will go from 5' to 3', and this will go from 5' to 3'. The direction of adding the nucleotides is always consistent, 5' to 3', regardless of the green part here. So even if this is 5' to 3', the addition of molecules or DNTPs will take place from 5' to 3'. And also over here, it is opposite, right? From 5, here it is 5 to 3, and here it is 3 to 5. But DNA will, the new nucleotides will be added in the same direction, 5' to 3'. So that's going to be consistent. Now, once the new strand has been formed, those two strands or two molecules of DNA will individually separate, just like denaturation. Like again, it will repeat itself. This was one cycle. This part was one cycle, and now they are repeating the same thing 30 to 40 times, so that small DNA sample magnifies or multiplies 30 more times, and you get that many DNA strands in the same mix. I hope that was clear. The advantages of using this particular method of PCR is that it is very sensitive, it is very fast, and it is very specific. So the primers that you use have to be sorry. So the primers that you use should be very specific to the DNA strand that you are amplifying. Its uses are in research, medicine, and in forensics. Next part is gel electrophoresis. Definition is pretty basic. It is a technique used to separate DNA fragments based on their size and charge. Smaller fragments will move faster and travel further than the larger ones. So this is the process, or just like basic idea of how it looks like. You have a container or a casting tray, that's what they call, and it is of glass with two ridges over here. This is the masking tape. You just put a tape on the sides so that the gel that you pour on it doesn't spill. Now, once it is covered, it's a box shape. Once it is covered from all sides, you pour in the gel, or agarose gel, that's the name of the gel, onto the same casting tray, and then put these combs. Okay, they have mentioned it over here. Yeah, you put this comb onto the recently poured gel and let it solidify for maybe 15 minutes or so. And once that is done, you remove the combs and you'll see these wells are being formed. It's going to be like a tiny well where you can load your samples. This is a micropipette. You use a micropipette to load very small, minute volumes, like maybe 1 to 2 microliters. And once your DNA samples are loaded, you will start the electrophoresis, which is provide the electric current, so that the DNA molecules will be like traveling from one side where they are loaded to the opposite side where they are attracted to. So now there is a direction. There has to be a specific direction to load this. So as we studied in the structure slide, that the phosphate molecules are negatively charged, and that's the reason DNA molecule has an overall negative charge. That is why cathode, which is the negative electrode, will be on the side where your DNA samples are. So you load your samples on the cathode side of the unit, and they will travel towards the positive side, which is the anode, when you turn on the power supply. Now, once this traveling has been completed, it should take about 45 minutes or so for the DNA fragments to travel. And then you can analyze these bands. So these dashes or these lines that you see over here are called bands. The pattern of these bands will tell you what are the fragments of DNA present in that particular sample. So the uses are checking the size of amplified PCR product. I forgot to mention that the first well usually has a ladder. Just like ladder is just a random name, but that's acting as a reference. So the ladder will have known size of DNA, and the samples that you load after that. So the first will be the ladder, and well number 2, 3, and 4 will have your DNA samples in it. Now you don't know what the size of your DNA sample is in those 2, 3, and 4 wells. So you are loading the DNA ladder in the first well so that you can understand depending upon where the band lies. So if this band is at a particular size, maybe like 250 base pairs, then you can know that, okay, these two bands are at the same location. So this band will also be of around the same size, like 250 base pairs. So similarly, you can understand what is the size of the amplified PCR product. Advantages are it is very simple, it is cost effective, and can separate DNA, RNA, and also proteins. Moving on to the applications of DNA analysis. Broadly, it can be used for forensic sciences, parentage testing or paternity testing, medical diagnosis of any diseases or genetic conditions, food industry, and agriculture. We'll learn about 2 of these applications today. DNA fingerprinting. It is a technique used to identify individuals by analyzing the unique patterns in their DNA. The process looks like this. You extract the sample, the starting sample, maybe like blood. This is a blood sample. You digest it. So restriction enzymes are the enzymes which cut your DNA samples into 2 to 3 different pieces. So the long DNA will be cut into 2, 3 pieces. So it's easier to observe on a gel, like electrophoresis gel. Now the DNA sample, which was a long thread, very long thread, will be cut into multiple pieces like this in the same solution. And after you perform electrophoresis, it will have different bands onto it. Those bands will be transferred onto a separate membrane. Once you have transferred it onto a membrane, you add or label it with a probe, which will detect the exact position of the bands. And when you observe that membrane or that membrane under an X-ray, you will have the DNA fingerprint ready for that particular person. Applications are in forensics, criminal studies, and paternity testing. So here's an example. At the crime scene, the sample that was found had these 3 locus points. Locus is a specific part on a specific portion of that particular gene. Now there were 3 suspects, and you have to guess which one is the actual culprit. So how would you do it? We have the reference over here. We want to see if it matches with any of these 3 suspects. You already have the answer over here, but to confirm, the 2 bands on this side have to match with the 2 bands on the suspect sample. So it's pretty clear and obvious that suspect 2 is the person responsible, because 2 bands of green, 2 bands of orange, and 2 bands of blue match completely with the suspect number 2. Advantages are it is highly accurate, even with small or degraded samples, it's going to do the job. And it is unique to each individual. Last is the PCR testing for SARS-CoV-2, which is the virus responsible for COVID-19. The molecular tests that they perform to diagnose or detect the presence or absence of the virus is by collecting the swab. We all know that. They collect the swab from nose or throat. They dip it into a solution and they will extract RNA from the specimen, which will be converted to DNA, because RNA cannot be directly tested like we can test DNA, or it cannot be amplified like we can amplify DNA, because RNA is single-stranded and DNA is double-stranded. That's why RNA convert it to DNA, which will be known as a special type of DNA known as complementary DNA. And once you have the complementary DNA with you, you amplify it. Just like you did with the regular DNA template, you amplify the complementary DNA into multiple copies, maybe 30, 40 copy cycles. After it has been amplified with specific primers, you interpret the results by doing the same analysis that we just studied, which should indicate the presence of viral RNA molecules. So they plot a graph, just like one over here. On y-axis, it shows the fluorescence, and on x-axis, they give us the CT value, which is also known as CQ value. Higher the CQ value, which means the cycle number. So as I mentioned, the number of cycles in a PCR reaction are ideally 30 to 40. So if your detection happened at a later number of cycle, if maybe it was at 35th cycle, then that means that your sample does not have that strong of virus. So they give the results in the form of a graph over here. The x-axis has the CT values or CQ values, which shows the cycle number, and y-axis has the fluorescence. Over here, if you see, the earlier you can detect, or the earlier the presence of the virus in your sample was detected, means that you have that high chance of being affected with COVID, or that many number of copies of SARS-CoV-2 is present in your sample. So having a lower CT value or lower CQ value is not good. Lower value means it is so much that it's been detected very easily. Now, if you have a higher value, maybe above 30, that's where the usual safe range is. You can still be positive with a 32 or maybe 35 CQ value, but you won't be that infective. So if you ever happen to see a PCR test report of COVID testing, check the CQ values or CQ or CT values. If the value is above 30, you can still be positive, but you won't be as infectious as others with lower CQ values. So that should be it about the DNA analysis part. Please send me an email if you have any questions. Thank you. Bye.